Hey, everybody. It's time for Q&A with A&V. I'm Vincent Racaniello, and with me is Professor Amy Rosenfeld. Hello, Amy.
Hello, Vincent. How are you today?
I'm very well, thank you. I've been thinking about viruses all day.
Let's not get carried away here. I mean, let's not start off with something that's like ridiculous.
How's everyone
okay.
tonight on this lovely live stream? There are almost 100 of you so far. Don't forget to click the like button. That's a finger up right below the video. So that helps to attract everyone else. And this is St. Paddy's Day. Happy St. Paddy's Day. And we'll get here a nice little set of shamrocks for that.
It's very good. Did you wear green today?
Well, no, I wore green yesterday. I messed up the schedule. I didn't get the email.
Oh.
Did you get email today?
No, I did not get email today. I decided to get dressed with, there was no heat in my apartment, so let's put on clothes that make me warm.
So
it's
Texas
not good
A&M,
when it's like 24 degrees out and there's no heat. Not a happy camper.
Texas A&M isolated the variant from animals, right? So they got it from the pets, a dog and a cat, who were the pets of a man who had COVID in February. And the pets are okay, apparently. And I'm not surprised. The variant is circulating, and it's going to infect animals for sure. Natalia writes, I would like to know if thromboembolism is a link to vaccine, AZ. No, I don't believe it is. I think these thromboembolic events were going to happen anyway. It's not linked to the vaccine. It just happens to be coincident. What do you think of that, Amy?
Yeah, I agree. It's coincidence. I think it's no higher percentage than in the general public. You need to do a lot more to convince me.
Yeah.
But I'm fussy.
After vaccination, if one then becomes infected due to exposure, what's the theory on one sustaining long COVID? Well, do you have any thoughts on this, Amy? I do.
Well, why don't you go first?
I don't. So we don't know the answer because no one has had this happen yet, right? But if you get the vaccine, if you have full doses, 1 or 2, whatever the vaccine is, you're going to be prevented from contracting disease. Now, we do know that
severe disease.
Mm-hmm.
Not it says nothing about asymptomatic. It says severe disease.
Asymptomatic. I was getting to that.
You said disease. So you have to be very specific about what you're talking about, because you said disease.
I did say disease, boss.
Right, and it only prevents severe disease and hospitalization.
Anyway, the long COVID can occur after mild infections. So what's going to happen with the vaccine is anyone's guess, but I think the vaccines will bring down reproduction enough to prevent long COVID. That's my guess, but it's a guess. Now, I'd be interested to see what Amy has to say.
Well, I think that you should start off with the truth, which is that we don't know that the vaccines do anything about mild disease. And I don't know what you said before. Mild disease. I don't even know what mild disease actually means. I think we should be very specific. We don't know if it prevents asymptomatic disease.
Well, there's some, as you know, there's some small numbers in the vaccines where they suggest that you're preventing infection, preventing asymptomatic infection.
Right, but nobody wants to put their money completely on that. That was a very small study. So the majority of the vaccines were defined by preventing of severe disease and hospitalization. The majority of long COVID cases come from asymptomatic, which I don't know how that qualifies as mild disease. So I don't know what mild disease is or not. You had a sniffle. I don't know. Is that mild disease and no sniffle is asymptomatic? I don't know. But so my guess is that if the vaccine actually does prevent transmission or infection, then you will not see long-term COVID. But if it just prevents against severe disease, then we will see long-term COVID.
Yep. Can an mRNA vaccine be made that has multiple types of mRNA in it, like a cocktail that codes for different proteins?
Yes, it
You
can.
bet. Multivalent, right? How many could you put together,
Yes.
Amy? 50, 100, 150 rhinoviruses?
Sure.
Yeah.
I don't see why not. Although you do have to agree that multivalent, at least for polio, multivalent was not as effective as monovalent.
Yeah.
So who knows if multivalent will be as effective.
That's right. I understand the second dose of COVID vaccine elicits memory boost, so more side effects. But why, if you get infected, will you have minimal symptoms? Isn't it a boost too?
I don't know that the side effects are from the memory boost or if they're from, your body really doesn't like the lipid nanoparticle and it makes some kind of effect against that. I never would have thought that memory boosts would make you more symptomatic.
I mean, here's the thing, Amy. If you get vaccinated, so you get vaccinated, like we got our flu shots. You get a flu shot, we had a sore arm, right?
Yeah, but they stabbed a needle into your arm. They harpooned you.
Yes, that's right. Now, if you get, let's say you got 10 COVID vaccine shots in the arm, do you think you'd get inflammation after each one?
Maybe.
Yeah,
The thing is you get multiple boosts of measles.
Yeah.
And I don't remember getting I got a measles shot in college and then I got one 3 years later when I graduated and became a technician. I don't remember being sick from the measles shot. So that would be a memory boost, right?
Yeah.
So I'm not sure that I'm buying into this idea that memory boost is causing all these side effects.
Yeah. So remember, when you're infected with the virus, it goes in your respiratory tract. So it's not very different from getting a shot in the arm, right?
Right. And many of these vaccines, yeah. I mean, yeah, I'm not clear.
2 a.m. in Gaza. Sorry you can't
Wow.
stay awake. You can go to sleep. It's fine. Okay, regarding H5N8, what triggers person-to-person transmission? Is it a random event? Well, these avian influenza viruses, when they spill over into people or any non-human, they don't reproduce well, and that probably impedes their transmission. But we really don't know why, and it's probably random. The right mutation or mutations have to occur to allow transmission, and at least for these like H5N1, it doesn't happen, which is good that it doesn't. Okay. Yes, sir. What's the latest with the New York City variant? It's, it's infecting people, but it's not the dominant strain. It's increased a lot. Did I show you this little quote, Amy, where the person who wrote the paper on the variant said, we just don't know if its expansion is due to random events or if it has to do with the virus. So,
I saw that.
It's being monitored. That's the best thing we can do. Cases in New York have plateaued, right, Amy?
Yes. They've actually, they dropped and then they plateaued, and I think they're dropping a little bit. But I don't think it has anything to do with the variant.
No,
it has to do with vaccination.
If I tested positive well after 2 doses, what type of quarantining do I need to do? Are you saying you got infected? That's quite interesting, isn't it? What do you think, Amy?
I'm not clear why you think it's interesting, but um, I tested positive well after 2 doses. So I don't know what I tested positive. What did you test positive for? Antigen? If you tested positive for antibodies against spike, then I don't know what it means because you developed antibodies against spike when you got vaccinated.
Well, if
If
you
you
got
tested
a
PCR positive for viral genome, depends on
yeah,
what the levels are, right? I don't know. Were you at 20 or were you at 42? I don't know what that means.
mm-hmm. Yep.
I need a lot more information.
Yeah, if the CT value was very high, if you did a PCR test, then you don't need the quarantine. Do you have any updates on safety data from vaccine trials involving pregnancy? I think it's too early yet, right, Amy?
I think so, but I did just see some blurb that said that I think some child was born with antibodies against the virus, and I think the mother was vaccinated. So I think it was like the suggestion was, you know, trans, you know,
Yeah,
transfer,
transplacental.
maternal transfer of antibodies.
Yep. In my country, they're vaccinated with Sinovac, but what if it fails? Can it be want to be vaccinated again? So
For sure.
you could, yeah. Why not? It's absolutely, you have Amy's permission and mine too. But yes, you could. If, for example, it turned out they did a study and too many people were being infected, you could get immunized again. Yeah, absolutely. Absolutely. Look at, hello, Professor Amy.
Hello.
Capital letters.
I know.
Oh, here's another one. I've heard about the new T cell test for COVID. They should send one as a gift to Amy.
No, they should just send me the money. They should just make a donation to the lab of a couple of, a significant amount of money. But
Amy made me laugh already. Love her.
it's hysterical. Somebody already complained. It's like, really? So, but that's fine.
Okay, the Tokyo seropositivity study from last summer has never been discussed here. A single company finding more than 50% of Tokyo had observed evidence of having the virus and no deaths. I'm not sure what those data are. I'm sorry. Do you understand what's going on there, Amy?
I have no idea what they're talking about.
We would be happy to discuss it, but one company finding that doesn't always mean that it's correct. It depends who they studied and so forth.
Well, I'm not 50% of Tokyo. Isn't Tokyo like millions of people?
Yes, many, many people. Many people.
I don't know how I don't know. I don't understand.
Someone in this forum linked to the Harvard preprint that indicated B117 increases the infectious phase. Yeah, so that was a study from the NBA, okay? It wasn't even a preprint. It was just a manuscript. Although maybe I looked at it a couple of weeks ago, and it's a very limited study. It looked like they shed by PCR a little bit longer. Yes, maybe 8 to 14 days. Not everyone did, but, it's not a really great study because it was a small number of people, and also they're all basketball players, so it's not clear how that represents the general population. But I'm looking out for the actual manuscript that comes out.
I mean, I don't really care what their profession is.
Well, if they're healthy athletes, that makes a difference, right?
Why does it make a difference?
You respond differently to infectious diseases.
Why? Are you not a healthy individual?
I'm not a basketball player.
I think like that's just a weak excuse. I think
that's what Christian Drosten said. Okay, I'm just.
I don't
channeling him.
I wouldn't repeat what he said.
Yeah, it's fine. It's fine. Do you know anything about Geert van den Bosch's claims? Do you know anything about this, Amy?
I don't even know. I don't know. What is his claims?
I don't know. Someone inform us, and then maybe we can comment on it. Question 2. Why don't we use daily antigen tests? Once confirmed positive with an initial to collect real-world data confirming the increased infectious phase. Yeah, we could. A lot of things we can do. And
Well,
some
you
of
have
them
to
are
have an
happening.
antigen test.
Yeah, we don't have an antigen test that is
And
working,
what
right?
are you going to do? Have an antigen test specific? I don't think an antigen test is going to be specific for one amino acid.
No, it's not. You're absolutely
So
right.
I don't know what I'm supposed to do with this information.
Well, you should do daily nucleic acid test, but that's going to be pretty expensive, right?
Yes. I don't
Question
see
for Amy.
anybody.
Here's a question for you. Do you feel your viruses have temperaments, like when they grow or they're happy and upset when the pH is wrong? Don't tell Vincent I asked. He does not like anthropomorphizing. I won't listen.
Okay. Do I feel my viruses have temperaments? No, my cells have temperaments. They don't like it when the pH is wrong. So then they die before my virus ever does anything. My viruses are very even keeled. But my cells are not that even keeled. So I treated today to look at DMSO for RD cells for this drug thingy. And I don't think my cells are all that happy so far in the 1% DMSO.
Okay.
I have a feeling neurons will be even less happy. Not that I'm going to sack some neurons to do this because they're expensive and hard to grow, but I'm thinking, yeah, not working out so well.
I love your opening monologue. So funny, everything great here in Boca. That's Amy's. Amy's got family down there.
I do. I don't like, you don't have to announce it. I wish to divorce some of them.
All right. Can we discuss CT values being hidden and does this new Britain variant really defeat PCR detection? Can you comment on that, Amy?
Are hidden? How are they hidden?
Well, they're not giving them to you. They're not giving them to you.
I thought that Daniel said that they were now.
Yeah, here now they are giving them out. So that's not, last year it was true, but now you can get your CT values. I got mine. Did you get yours, Amy?
No.
And the Brittany, explain the UK variant and PCR, the dropout business.
So the dropout business has to do with the location of, I guess, of where one of the primers annealed to generate that specific band. And when you have when you alter or you're missing pieces, the primer cannot anneal properly.
Right.
And then it does not amplify that piece. And so you get no signal. And then when you run a gel, it looks like the band is missing.
Right.
So I think that they've changed the test subsequently. No.
Yeah, they think they have adapted it to get the right primers for sure. Yeah, absolutely. Yeah. Let's see. Where did the virus come from? Wuhan Institute of Virology? No, probably not. Most likely not. It came from a bat, and we're going to try and find it. People are going to go and trap animals and find it, and maybe in a year or two they'll find it, which they did for SARS-1. They found the animal with 99%.
Took like 10.
What's that?
Took like 10 years.
Yeah, it could take a while. But
well, it's not even clear that it's from a bat that's near there, right? Yeah. I mean, according to Lympha's paper and the paper by the guy whose name escapes me at Tulane, there are bats in a 4,300 square kilometer distance from Japan all the way to Southeast Asia that harbor these viruses. So, and people move, and live animals get on trucks and move various countries. So it's not clear to me that, you're going to just, plop down in a spot and pick it up. I mean, it's out there, but it's going to take you some time to find it.
Yep, we'll find it. Ontario Science Table has been publishing reproductive numbers for the variants 1.35 versus early. Doesn't this suggest a subtype with unique properties? No. I will tell you why. Here is why. Here's the formula for reproductive number. Can you see that, Amy?
I see it.
Reproductive number is tau times C times D. Tau is the probability of infection given contact. C is the average duration of contact. D is the duration of infectivity. You can see C is a human factor. So changing R naught is not just dependent on the virus. It's also dependent on human behavior, and nobody's talking about that. Well, some people are, but no one pays attention. So not necessarily the virus. It could be, but can also be the way people are behaving. I mean, I frankly think the increases in Italy and Germany are because people are misbehaving. They're not wearing masks. They're not separating from each other and so forth. Can the LNP technologies be used to encapsulate potential antiviral candidates that function better inside cells? What do you think of that, Amy?
The lipid nanoparticle?
Yeah.
I mean, yeah, in theory, you could put in antivirals, but it's not clear to me that that would be an efficient way to deliver drugs.
Yeah. You know, the best way
and you're not even, so you're not, it's not clear to me that these lipid nanoparticles can go down the respiratory tract to the appropriate location.
Yeah, right.
It's very different than stabbing and harpooning yourself into your deltoid.
You like to call it harpooning, don't you?
Well, it is, right? They go... It's not my fault you weren't creative and think of it as a harpoon. I mean, that's what they do, right?
What's your view of kids and COVID? New trials are starting for kids. So you think the vaccines will be safe? You think it's better for kids to get infected or hope to get vaccinated?
I think we fucked up kids and COVID from the very beginning. I think we will find the vaccines are fine for kids. And I personally, though I have no children, don't think I would want to risk having my child infected even if the quote unquote disease is asymptomatic, with the potential of having long-term COVID for the rest of their lives or however long it lasts. I wouldn't want to take that risk. But I think we have nobody but ourselves to blame, because for some reason we lost some reasonable thought at the very beginning.
Yeah, I agree.
So I'm not clear why we did this.
Daniel talked about long COVID being more frequent now in young kids. Yeah, that's totally right. You absolutely nailed it.
I just don't understand why you would come out when every other respiratory disease comes from children and they bring it home to parents. Why you would come out and say children don't get disease, and then children don't transmit disease. It seems to me like that's the wrong position to have ever taken. I mean, if we were to do an experiment on a new virus, right? The way we model our experiments is we say, okay, EV68 is a picorna, it's an entero, it's going to function similarly. We plug it into our model and we assume it's similar until we find the difference, right?
Mm-hmm.
Why would I do it the opposite way?
I agree with you. I totally agree.
Just don't get it.
People are reporting long COVID getting better after vaccination. Is it placebo effect?
No, Daniel's tweeted this, and I think Daniel's probably going to talk about it tomorrow with you. No,
I don't know. I don't get to hear what he's going to talk about, but we'll see. Good.
But yeah, it's not a placebo effect.
Before Amy, plaque assays and organoids were not in my vernacular. If I suspect I may have contracted COVID-19 a while back, what's the best test to confirm that or rule it out?
Presence of antibodies.
Antibodies. Go get an antibody to N. Once COVID is over, do you think people should routinely mask to stop the thousands of yearly flu deaths? That's an interesting question. You could just get vaccines, right? You could get vaccinated. In many countries,
thousands of people die in the United States after getting vaccinated. After, with a vaccine.
Are you sure about that?
Yeah, I'm positive.
Well, maybe this will impress upon people the value of wearing masks. But in many countries, they do in flu season, for example. I think
I don't think that Japan has anywhere near a flu problem like we do.
Yeah, it's not a bad idea, but it won't be mandated. It should be suggested by the CDC, right? They should say, we recommend mask wearing since you're used to it now.
Well, nobody said we were used to it.
I'm used to it, aren't you?
We can't judge by us. We're a very small minority.
Yeah. Amy is sitting too close to that giant biohazard waste can.
Well, I'm not that close, and I greatly appreciate their concern for my safety, but it's fine.
If mRNA vaccines are created in a cell-free bioreactor, has the technology advanced enough to replicate viruses without cells? Only one virus has been shown to reproduce without cells, and that's poliovirus, done by Eckhard Wimmer years ago. So it's very hard to do. Most viruses, you need this. Well, there's something in a cell that you need, the structure, the arrangement. It doesn't work.
Well, he didn't exactly make it without cells in a bioreactor.
Well, he cracked open HeLa cells, right?
Yeah, he took the cytoplasm of HeLa cells and he programmed it to make polio.
Yeah.
So it has all the crap that a cell that's in the cell cytoplasm there.
Mm-hmm. Yeah.
Right? That's not what a bioreactor is. Right. That's not what a bioreactor is.
Yeah, I don't think we can make viruses without cells, especially not on scale.
Well, I'm not clear. Yeah, I'm not clear how that would work.
Very high number of cases in Brazil. Some say P2 variant not proved yet. My guess is fitness and the lack of care and distancing. I agree. I think it's a combination of a little improved fitness on the virus and people relaxing. Like you say, almost everybody living normal here. Yes, I totally agree. And people seem to leave that out.
Oh, you want people to take responsibility? That's not acceptable policy.
Yeah, so John Snyder says, Barry's correct. This has been reported after one dose of vaccine and sequelae improved slightly, covered a week ago. Really? Okay. Since our pets can get COVID from humans, wouldn't vaccinations for pets be a good idea? What do you think, Amy? Should we vaccinate pets?
Well, they're not dying.
Not yet. There haven't been that many infections that we know of, right?
Yeah, I mean, I guess at some point it would be fine to vaccinate pets.
Yeah, because pets get a lot of other vaccines. It's fine.
Sure.
That's a good point.
I think we need to first probably finish vaccinating humans.
Opinion on why Florida having a low number of deaths considering it didn't lock down. Is that correct, Amy?
I've read stuff that says that, yeah.
I don't know if you can really figure it out. We don't have the data. Every place is different. I always tell people, don't compare death rates in different countries, different states. Every situation is different. You know, the UK variant is spreading around Florida. And so what does that mean?
Yeah, but this is more like people are so you're not supposed to get political, but there's a certain realm of this population in the country that says, look, New York got destroyed by the wacky blue people and their death rate is huge compared to Florida. And this is why you don't need to have mask mandates and open up and stuff. However, one has to recognize the fact that New York's pandemic occurred in March when we knew very little and we didn't even know how to manage the treatment for patients and whatever. Whereas Florida occurred like 5 months later when we figured out how to manage things, and we had more rapid tests and stuff. So what are you telling me? You're telling me that when we actually have things that we know how to use, we can prevent death? Excellent. And for the next disease, we'll have the same thing. We'll have to learn the same lesson all over again. I don't understand.
Here, let me show you another image. So this is the case fatality ratio from early in the China outbreak. Here's Wuhan in January and the rest of China. And here's Wuhan a month later. The death rate goes way down. So as Amy said, you figure out how to deal with it. And that's why.
I mean, if you look at it in 1920, probably like any car accident you had, the people died. Today, very few people die from car accidents because we know how to manufacture cars. We have airbags, we have seatbelts, we have blah, 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 blah. Just don't understand.
Got my first Pfizer shot 8 hours ago. Doing good thus far. How long for me to build up antibodies?
Minimally 2 weeks.
Yeah, good job. Good job. If I understand correctly, lack of side effects of a vaccine does not mean you're not protected. Correct. How do we know? Because irrespective of having a side effect, people make antibodies. They do studies. And for example, if you look at the phase 1 and 2 results, they ask people, did you have pain at the injection site? And some people do and some don't. And you can see who made antibodies, and it doesn't matter about pain at the injection site. You still can make antibodies. All the data are there to look at, actually.
I never understood why people made these analogies or correlations. They're not analogies. They're correlations. I just don't understand.
Texas, they're training dogs to detect COVID-19. Okay.
It has a smell? So is it Chanel number 5?
I think it's the smell of the infected people, right? They're putting off volatiles that the dogs can detect.
Oh, I was so hoping it was Chanel number 5.
You like Chanel number 5?
No, but it's just very famous.
Do you think it's safe for a nursing mother of 10-month-old to get vaccinated?
Yes.
Yep. It's not a problem to pass antibodies to your 10-month-old.
It's good.
With respect to neutralization assays and variants, how good are pseudotypes versus other approaches? I mean, there's different, like who said recently, Matt Freeman said there are differences between pseudotypes and infectious virus neutralization assays, but he thinks in the long run they all track the same. What do you think, Amy?
I guess that they track the same. I've never done it, so I don't really know. However, I do believe that I pointed that out during a podcast on Twiv last year, or at the beginning of this year, when I said, pseudotypes and infectious wild type virus are very different because we don't, pseudotype, you're just expressing spike. Maybe it's completely in the right orientation and grouped as trimers, or maybe the need for E and M, which we don't understand, is required. Who knows?
Yep.
I believe I pointed that out.
Why did the people of India, where it's so crowded, have so few deaths? Now, we've brought this up before. We really don't know. Do you have any insight, Amy?
No.
We could speculate, but we really don't know. And no one knows. If someone tells you they know, they're not telling you the truth. Have you heard the story out of Atlanta saying loss of taste and smell could be a sign the virus could be hiding in the brain? I haven't heard it, but it's bogus. That's not right. Loss of taste and smell is from reproducing in the cells lining the respiratory tract that are involved in taste and smell. It's not going in the brain. Right, Amy?
Well, they're not lining the respiratory tract, right? They're in the olfactory epithelial that maintain the healthiness of the neurons, right? That's where the virus is replicating. And then the neurons don't work properly, even though they're not infected. This is my problem with a certain individual's work, because she screwed it up.
I know who
And
you
then she
mean.
continues to propagate, to screw it up.
I know who you
And
mean.
then, yes. And then today I saw a preprint, or very early this morning. I got so mad I had to go running at like 5 a.m. Where they took iPS cells and made different cell types of the brain. And then they compared it to a neurotropic flu virus. And they say, no replication, no replication, no replication, but replication of flu. So once again, there's no repercussions for the person who screwed it up.
17 nursing home residents contract COVID after being vaccinated. Some one dose, some two. Thoughts? This is hearsay. Maybe they got infected by the person who came in and vaccinated them. They got infected the second day when they wouldn't have protection. I don't know. There's always a solution, and getting it from the news is not it. So until we hear more, I don't want to speculate on that. In the beginning of the pandemic, there was an estimate that 10% of transmission was via hands. Any update? Well, in the TWIV that's going to drop at midnight tonight, you'll hear that in a survey done in San Diego, about several thousand swabs of surfaces, only 20 were positive for fragments of RNA. So I'd be surprised if it were even that high. Second, Moderna today. There's contrary information about taking NSAIDs after vaccine. Could they adversely affect the immune response and effectiveness? I believe Daniel has said go for it, right?
Yes.
NSAIDs are fine. Not a problem. Not a problem. Oh, Amy will know about this. There's chatter about possible cross-reactivity of antibody cells for other coronas to 2 months ago. Any updates from recent research? Know anything new, Amy?
I don't. I think that there is cross-reactivity, but it's very minor and it's not neutralizing.
Should we ask Susan Weiss on Friday?
Yeah, you can ask her. I mean, this isn't really what maybe she'll know. I mean, this isn't really what her interest lies in and where her expertise lies in. This is like conflicting work by Bloom's lab and somebody else's lab. And I believe that Jesse doesn't think that there's any cross protection. And I'm going with Jesse. I'm going with Jesse's opinion.
Is anyone working on a real-time, non-invasive, accurate testing? Spit on this, like pregnancy test.
Why?
Well, someone is working on it. I know at least one company, but it's not out yet.
Why?
Why what?
We're 14 months into the pandemic. 15% of the American population has been vaccinated. What am I going to do with this test now?
Oh, you can talk to the companies, but they're still doing it.
Well, I mean, the government just released like $10 billion to test schools, to have a school testing program.
Yeah.
Okay.
Well,
I'm not really clear what we're testing for now.
Okay. With the UK nursing home COVID deaths and people already vaccinated, we're sure the pandemic will end. Yeah, it's going to end. I think this is a bogus story, and we're not getting all the facts. That's all. John wants to organize your lab.
Okay, come on. I don't organize. I'm not a good organizer.
J&J announced the vaccine can be used with pregnant women, as I'm watching now on the news in Spain. Did you hear about that, Amy?
No.
Thank you, Jeffrey, for your super chat. Much appreciated. And thank you, Rita, for your super sticker. Much appreciated.
What's a super sticker?
If you look at the real chat in YouTube, they have little like emojis, basically, cats and stuff like that.
It's great. Lemon character doing victory dance.
Yeah, so see, it's being described here because it doesn't show up on the on it, but lemon character. Some of the COVID. Hello, Frank. Some of the COVID vaccines target spike. Would COVID vaccines be effective on other coronaviruses?
No. Although I did just see a paper by Barrick and colleagues that are trying to make a pan-corona vaccine.
2 weeks after I get my second Moderna, would it be safe to ride the bus and volunteer again? Yeah, I would wear a mask. I got my second shot over a month ago, and I still wear a mask, and so does Amy. So I would do that. Tokyo, 13.5 million people. There you go. Would the
What?
newly approved T-cell test be able to differentiate between natural infection and vaccination?
If it looks for things that are, if it looks at T cells that are derived, that are specific for like N or if other proteins besides spike, sure.
Yeah, for sure. Oh, Rita's working on the coloring book. That's cool.
It's great.
Yes, Amy, you're going to be in it. That's very good.
Great. I'm looking forward to seeing this book.
You like coloring books? Oh, Gert. Here's Gert. Gert is making rounds saying vaccines will destroy your immune system. Well, nothing could be further from the truth. My immune system is great. Amy's is great. I know many, many people who've vaccinated. That's just typical anti-vax stuff. So forget it. It's not even worth talking about. Is it correct to think that in Brazil, as we carry out an insufficient amount of testing, we cannot say the increase is due to the new strain of the Amazonas? Well, you can, if you do 100 tests and they're all the new strain, you can say it looks like it's most of the infections, but you need to know exactly where it's going and if people are behaving differently. That's what I said earlier. It was a study that detected viral RNA up to 3 days on fomites, where there are studies done to detect virions. So the first study out of the Rocky Mountain lab last week, yes, they looked for infectivity. And they could, but first of all, they put 1 million PFU on the surfaces and in a laboratory condition. And they found a few days, infectivity would endure a few days, but not all of it. It has a half-life, right? What percentage of the U.S. needs to be vaccinated to end the pandemic?
Believe 70.
70%. And maybe infections would contribute to that. And we're underestimating infections. So we may be there before we know we're there based on vaccine frequencies. How do we donate to Amy's lab? We did a fundraiser a while ago, remember?
Yep.
We could do another one and announce it.
Yeah, they can still contact the crowdfunding people at Columbia and make donations for sure.
We will pass on the information next time.
For sure.
Can someone be vaccinated while on antibiotics? I don't see why not. Do you?
Yeah, should be fine.
Do American scientists throw AstraZeneca vaccine under the bus often? No, I don't trash it. I think it's fine. I think some of the testing has been weird in England because they had the half-dosed issue and in Brazil, but it's in phase 3 in the U.S. We'll see the results. I just don't know why they didn't use a prefusion stabilized form. I'm curious about that. But I don't throw it under the bus.
2 more questions, and then I have to add overlay.
Okay, conclusions and relevance. COVID-19 it may have spread widely across the general population of Tokyo. Despite the low fatality rate, herd immunity may be implicated. What are you saying, that he reached herd immunity before? I don't think that would explain it, before fatalities occurred. What do you think about new strains in the vaccines? I think the vaccines so far all protect against death and hospitalization against the variants. And as Amy said earlier, that's what's important.
Well, also Crade's paper and Seti's paper says that the T-cell epitopes haven't changed.
That's right. The T-cell epitopes haven't changed. And Alex Seti will be on TWIV in a couple of weeks. So stay tuned. One more, Amy?
Mm-hmm. I don't. And so while he thought it was a boring paper, I think that okay, maybe it's not the most exciting scientific result or whatever, but it's actually a significantly important result. Not all
Yeah.
important results are, earth shattering.
Apparently, commercial labs run all PCRs to the same CT41. This seems to be a useful indicator for how mild severe illness will be or the direction of illness. What do you think about that? You're the CT guru.
I don't see how it's a useful indicator of how mild or severe
Yeah.
illness will be or the direction of illness. So the direction of illness you can only determine if you're taking multiple samples from the same person. Hopefully sequential days, maybe. There is no suggestion to my knowledge. Maybe you have new knowledge. I don't know. But it doesn't seem to me that when you have a CT value of 11, you're always going to be hospitalized and have
No.
severe disease. And when you have a CT value of 23, you only have, the sniffles. So I know you like to think that there's a correlation between viral load and HIV and the development of AIDS, but if you don't have virus present, you're not going to get disease. So
Thank you again, Amy.
thank you. Oh, we need to change the liquid nitrogen tomorrow.
Will do. See you in the
I
morning.
forgot. All right. Have a good
Bye.
evening.
Thank
Bye.
you for joining again.
Bye.
Bye. Okay. Let's go. I'll stick around for another 45 minutes or so. COVID GI symptoms seem common since our tests target the upper tract. Are we flying blind? I'm not sure that these are GI infections, frankly. You know why? It's very hard to isolate infectious virus from the gut. Now, there are some papers that suggest reproduction in the gut. I don't know that it's that extensive. And I don't think that we need to be diagnosing it, say, from fecal swabs or something like that. I think the respiratory tract is the main site of illness and virus reproduction. So, look, the saliva tests for antigens are a good idea. Amy's argument is we don't need them anymore, but people are still working on them. And I have a contact at a company working on them, and he said it's very difficult to get them to work. So there you go. This is a better time. 8 p.m. is really not a good time for you. It'd probably be better to do it earlier in the day. I mean, I'm considering doing another one at a different day of the week earlier for people who, have weird time zones. So stay tuned. Stay tuned. People are reporting that their doctor claims they've had corona infection at one point if they experience vaccine-related symptom, headache, joint pain. No, you can't diagnose this symptomatically because it's the symptoms, right? Headache, joint pain, etc. is too similar to other viral infections. You have to have a diagnostic test of some sort. Yes, here in Switzerland, there's no such thing as an EUA. After review, Swissmedic approved Pfizer and Moderna, but not AstraZeneca. Okay. I think that's fine. They can do as they wish. I don't know. Maybe the AZ won't get approved here, but the FDA only uses data from the U.S., and that's why they're doing the phase 3 here. When infection spreads from the upper tract to the GI, is the incubation period another 4.5 days? No, basically what happens is you're swallowing mucus because mucus is being cleared from your tract. You swallow it to get rid of it, and the virus goes into your GI tract. Some of it survives the low pH of the stomach and so forth, the enzymes that are present there, and ends up in the stool. Whether it reproduces is not clear to me. I think it's very inefficient reproduction in the GI tract. Do you know the philosopher Daniel Dennett? I do not. He says it can be justified to talk about designed systems as having goals, whether designed by us or evolution. That's fine. I have no problem with that. The reason I don't want people to anthropomorphize viruses is because it makes you think of them in ways that are not appropriate because they don't have human-centered goals. All right, they don't feel as we do, they don't think as we do. So that's the only reason. And, if other people do it, that's fine. But in my course, I tell my students, don't do it because you're ascribing human goals, and that's not appropriate. That's the only reason. Michigan's case rate has gone up dramatically. This is in the face of 25% vaccination, another 33%. I don't believe these numbers at all, okay? If you really have 60% immunity, the cases are not going up. Something's wrong with the numbers. But, if the people who are not immune are hanging out together, community grouping is a big deal with a disease spread, then it's going to spread in them, irrespective of herd immunity. So there's always something. Excuse me. Sorry. Do I really believe dogs can sniff the virus? Well, what I believe doesn't really matter, right? It's the facts. And some reports have suggested they can smell the scent of people who are infected, not actually the virus. No, I do not believe the dog can smell the virus. If you held the tube up, no, but maybe an infected person gives off organic vapors that smell a certain way. Sure, dogs have amazing senses of smell. Yes, according to NPR the other day, WHO is looking at animal farms in China. We'll see what happens there, right? Thank you, Post Polio News, for your super chat. 5-part question following. All right. Does Amy have any plans to share any lectures or videos? Not that I know of. Amy is really devoted to her lab work. She needs to do that to get established in the field. So you're not going to see that happening anytime soon. It's possible it started in bats. Was it taken to WIV where they did gain of function? No, it is not. Nobody had anything close to this virus. So you could say maybe they did, but that's an assumption. That it's not based on any facts, whereas we have bat isolates that are quite similar to this one, so it makes it clear that the ancestor is somewhere out there. I mean, you could make up anything. You could make up that people got sick in November, sure, but we have the sequence. That's where I don't get it. As virologists, what keeps you awake at night? That's a good way to put the question, right? Because there are a lot of things that keep me awake. It's not all virology. Well, unfortunately, it's keeping our lab funded so we can do research. I worry a lot about that, and I know so does Amy. And I really do worry about being prepared for the next outbreak. In this pandemic, what has kept me awake is this knowledge that this could have all been prevented, and it would not have had to leave China, really, if we had had antivirals or vaccines, and we didn't because we were unprepared. And I hope we're prepared for the next one, but I'm not sure we will be. I think when it's over, people forget, and the research that we need to do is not going to be supported. So it's a real problem. I have comment about AstraZeneca vaccine. So I think the people who had blood clots were going to get blood clots whether they were vaccinated or not. As far as I can see, there's no evidence that the clots were caused by the vaccine. We hadn't seen them before. I just think this is another example of people having things happen to them. When you immunize millions of people, you're going to see those events, and they have nothing to do with the vaccine. And so it's too bad that they had to stop vaccinating people. I'm sorry, Post-Polio News, I missed all of your questions. I'm so sorry. Let me start with one. The following question is not about AZ, but rather about alleged statements perhaps not being as informed as they should be. According to this newsletter, this professor at Witwatersrand and lead researcher of the South African trial, now I'll get to 3, says there remains a likelihood they will prevent severe disease since they rely on the same vector as the J&J. Well, it's not the same vector, right? It's similar. It's AstraZeneca is a chimpanzee adenovirus, and J&J is a adeno 26. So they're both adenoviruses, so you could split hairs that way. It's a mistake not to use them. Yeah, so this is Chadox, ChimpAd, and J&J human. Can they be considered the same? Of course not. They're not the same, but they're the same family of viruses, right? They're adenoviruses. But I don't think that's really the relevance here. You know, the immunogenicity, the durability, they could differ. Different isolates of viruses can have very different properties, for sure. In part 5, if comparing the two vaccines, surely J&J prefusion stabilized are more significant dissimilarities in the vector. Absolutely. That's the one difference that I question. Why AZ is using not prefusion. I don't get it. In Germany, B117 is now 50%. Do you have a percentage for California? I think it's 20 to 30%, the last number I looked, but there's competition with the California variant, so that's keeping it down, I think. These variants are competing with each other. Is spike prefusion the shape of the S before it binds to ACE and changes shape? Yeah, when it binds to ACE, then it extends to fuse. It goes into the fusion conformation. You want to see a picture of it? I have one here, I believe, readily handy. Actually, I don't, but I'll show it to you anyway. It's nice to show some visuals, right? Here we go. So that's the spike. This is prefusion conformation. So the cyan down there, that's the fusion peptide. And in the post-fusion, it shoots up to get into the membrane. And so that is why you stabilize it in the prefusion conformation. Absolutely. Frozen food is being named as a source, being pushed by Dashak. Is that a joke? I don't think frozen food is a source. There's just no evidence that food is responsible for transmission. This is, yeah, I don't agree with that. I'm sorry. I have a colleague in Germany in lockdown, but attended the meeting today with 30 staff members indoors, no mask. Yeah, thank you, Hoplite76. Of course it must be the boogeyman variant. Yeah, people are perfect in their behavior, right? Yeah, it's mostly people. It's not the variants. It's not the variants. Okay, yes, every kid brings disease home to the family. Why would COVID be different? Totally. I know Amy said this a while ago, but that's the delay in the chat. I'm sorry. I never believed that for a minute, so I kept my kid home and still homeschooling them. Yeah, kids get infected, and now we know they get long COVID. And you know, Daniel Griffin said last week, the worst thing is to tell the parents that their kids have long COVIDs, and the parents saying, I thought it wasn't a serious disease in kids. What do you say to the parents, right? And yes, I do think people wanted to push schools back open. You're absolutely right. It had nothing to do with the virus, right? I mean, kids should be in school, I agree. But to ignore the science or to selectively pick science that says your agenda, that's just not right. Oh, look, Alice, then each of these had new strains. The strains were circulating before the vaccine was tested. So don't jump to conclusions, please. Where did the idea that kids aren't affected come from? All right, so there are two things. There's affected and infected. So early last year, some testing showed that kids weren't infected that much, but they were only testing symptomatic kids. And it turns out that more kids are asymptomatic than adults, and so they were missing that. And when they did subsequently serology, they found that a higher fraction of kids, in fact, were infected. And then they didn't seem to get as sick. And it's true to this day, they don't have the severity of the disease in adults, but they do get long COVID. Long COVID can happen to anyone with asymptomatic disease and up. Is it possible the currently observed effectiveness of the vaccines are artificially elevated due to increased population immunity between time of trials and rollout? Bingo, you hit it on the nail. Dojo Create. I've been thinking about this a lot lately. So we vaccinate people, and then, you know, in the next month or two, we check to see if they're protected against disease. And at that time, antibody levels are really, really high. And if you waited 6 months to a year, the levels would be really low. So you would get infected, then your antibodies would kick in 2 or 3 days, but you would get infected. So I think, yes, the vaccines are giving higher efficacy than, or maybe not against disease, but against infection. So I think it'll be interesting to see how this pans out going forward, for sure. My mom is getting the Pfizer shot. She's nervous. She'll be fine. Most people are fine. Not an issue. But it's okay to be nervous, but tell her that many people have gotten it and they are fine. What options are available for amplifying nucleic acid sequence without knowing if you don't have primers? Well, you can use random hexamers. So you can get these and they will basically prime anything. And we use those a lot in the lab when we don't have a sequence to make a primer to. So you can do random hexamers, are what they're called. Well, the mRNA vaccines are transcribed in vitro. They're not actually chemically synthesized, right? You know, an in vitro transcription reaction, Tom, you must know what that is. So you take DNA encoding the spike, you add an enzyme to make RNA in vitro. We do that all the time in the laboratory. Could you make bacteriophage using mRNA to have a reliable way to make them? So, yes, you can take a DNA copy of the bacteriophage genome. You could transcribe it in vitro using an enzyme like we just said for the mRNA, and then put the RNA in cells, in bacteria, of course, and out would come phage. Absolutely. Absolutely. Do you think that what we've learned from this pandemic about COVID vaccines will make future influenza vaccines better? Yeah, I do. I think people are going to try mRNA vaccines for influenza right away, and that could improve it right off the bat. You know, instead of having to make them in eggs or even in cell culture. So yes, I do. I mean, I think vaccinology in general is going to be improved because we now know we can make a vaccine in less than a year, multiple vaccines. How about that? That's just amazing. Can we encourage healthy sleep habits to increase vaccine efficiency? You know, people have trouble sleeping. You could tell people you need to get good night's sleep, but sometimes it's not easy. You know, I go try and get to sleep by 11 and I wake up at 3, 4, 5, can't get back to sleep because I'm thinking about things. So it's not, I mean, I agree. It's probably good to get a good night's sleep for a good immune response, but it's hard to do. Does Novavax have increased chance for diversity of epitopes? I don't think so. It's a protein, right? It's a purified protein as opposed to making the protein in you. So I don't think that there's an opportunity for that. Are the identified variants based on changes to the envelope spike, et cetera? Are they sequencing? So they sequence the whole genome, and there are changes elsewhere than the spike, for sure. For example, ORF8, which is a very interesting protein that's involved in modifying suppressing immune responses. That has changes, but nobody talks about them. All people talk about is spike. They're myopic in that way. Oh, spike is all that matters. But yes, there are changes elsewhere, and they could make a difference. For example, the ORF8 deletion could make the virus less virulent, and that would make it transmit better. Nobody's thinking about it. We have this hive mind when it comes to genome mutations and what they're doing. And I just wish people would think outside the box, think more openly about what they're doing. It really bugs me. Does developing severe symptoms correlate to higher chances for long? No, not at all. You can get long COVID from moderate symptoms and maybe more likely to do that. Yes, if you vaccinate pets, then they shouldn't be a reservoir, and that would be good, of course, right? All right, my mom asks me if she should get the Pfizer vaccine or any other COVID, asking me what are the benefits and risks. What should I say? Well, so the risks of getting COVID, depending on your age, right? So they go up substantially after age 60, 65. And there are wonderful tables that show that, but you don't need to show her tables. You say the risk of COVID, of you dying or getting severe disease from COVID is very high, and there's very little risk from the vaccine, and it will prevent you from dying from COVID. So that's the risk-benefit ratio. And millions of people have already gotten these vaccines. And so if you want to wait a little longer and see 100 million people are vaccinated, that's fine. And no problem with that, but be careful in the meantime, right? Are there research findings on COVID effects on children's development in utero when mother is symptomatic? Very good question. I'm not aware of any. No doubt it will be looked at because people obviously are infected under those conditions. Okay, what do you think of the study in Nature using mice, hamsters, and ferrets saying that D614G enhances replication and transmission? I think the data are straightforward. Whether they apply to people or not, we don't know. But I think what they're saying is that this change improves the fitness of the virus, its ability to reproduce in the host, and it could be responsible for it dominant. So D614G is now in every virus that you isolate, right? Since last year sometime. And when it first came out, the claim was it was more transmissible. I didn't agree because we didn't have the data. Now, data like this are more in line with that, with the caveat that no one has done a human experiment that would address it, which is very simple. And now you can't do it anymore, but they should have done it back then. Take nasal swabs from people with or without D614G and see if they make more infectious virus with D614G. All they did was PCR and say, oh, yeah, there's higher RNA. I don't care about higher RNA. I want to see infectious virus. And that's a problem. People are not adhering to virological standards. Have we seen serious spillover from pets versus farmed animals? Well, we don't really look that much, but in the Netherlands and Denmark, there was big time spillover to minks, and then they had to cull them to stop that. And I think here in the U.S., we're not even looking that carefully. We pick up the random pet here and there, but it could be more extensive than we think. Could be going into rodents, wild rodents, for example. So I think that's a reality, and it's going to establish a reservoir if it's true, and we will always have to vaccinate against this virus as a consequence. How many documented cases are there of COVID reinfection? So here's the thing. The bar is very high. If you want to prove a reinfection, you need the genome sequence of the first virus and the second one to show they're different, and it's not just the virus hung around in you for a while, because it can do that in certain people. So very few, less than 20 cases have that kind of documentation. But as Daniel Griffin says, it's still very rare. If you are immune after an infection or vaccination, you're not going to be reinfected. If you are, it's because you didn't respond. And those responders are very rare. What do I think about Sinopharm and Sputnik? I think they're fine. The phase 3 are out. We talked about them on TWIV. They look good. They're safe. No adverse side effects. You get good immunity. They protect against serious disease. And so I don't know what else you would want in a vaccine. So I think they're all fine. I don't see a vaccine that I think that has gone through a phase 3 in the U.S. or elsewhere that I think that is not fine. Absolutely. Will you please try to get Chantal Abergel and Jean-Michel Claverie back on TWIV? Yeah, it's hard. It's hard to get them back on. The time difference and so forth. They're always fun to talk to, for sure. Are what we call a virus actually exosomes? There's some. I mean, they're little vesicles, but not all viruses have membranes, right? Poliovirus has no membrane most of the time. Parvoviruses, adenoviruses. So it's not a generalization, and there are differences. Any thoughts on Novavax? So, The preliminary phase 2B data came out a few weeks ago. They look pretty good. I'm waiting now for the phase 3 from the U.S. They should be out. Matt Freeman said they'd be out in April. I think it's a good vaccine design, and it's different. It's pre-fusion stabilized. It's adjuvanted. I think it's a good vaccine. A lot of people talking about Florida, but I don't want to dip into that. Do you know if children who had Kawasaki have higher risk for MSI? No, I don't know that that's the case. I don't think there's an increased risk. My father had first shot on his 81st birthday. Congratulations. I'll send some good nurses to avoid harpooning. I didn't feel it was harpooning, but yeah, that's a good way to look at it. Are they bacteria that eat viruses? Gosh, I've thought this has come up before. Not technically, no. Viruses infect bacteria, but and the ones that can't, do they eat them? Not that I know of. Some infectious disease specialists think the next pandemic will jump to humans in consolidated feed operations. More animals, more spread, more mutations. Hey, that picture of the snow ice sculpture thingy, I used that on one of the TWIVs. Cool. I don't know about consolidated feed, but certainly going to come from some kind of animal, non-human animal for sure. Pfizer wants to jack up the price on the vaccine. Of course, Pfizer is big pharma. They want to make money. And I'm very unhappy with some of these companies. I like Moderna. They used federal money to develop their vaccine. They're forthright. Pfizer is secretive. The vaccine is fine. Don't get me wrong. Not happy with the company. Oh, a French virologist said it's all BS here. Politicians in the past were paid off by a local farmer to push the HPV vax. No, HPV vax is actually a terrific vax. I hate to burst your balloon there. Really good vaccine, and its sterilizing immunity is induced by it. One of the best vaccines out there. So that's all BS. Sorry. Let's see. A lot of India, a lot of Florida. In severe COVID, do we have an idea of the portion of organ damage caused by microclots versus damage by excessive immune response? No, we don't. It's a mix, and it depends on the patient. But I'm glad you say that because it's true. Microclots versus excessive immune response. It's not typically virus destruction of tissues. Could it be possible there are proportionally more asymptomatic infections, hence less testing? Where are you talking about? I've lost track of this thread now. I'm very sorry. Very sorry about that. You have to remember that the difference is country to country, state to state. There's so many variables and things happen different. It's really hard to draw conclusions about what's going on. And, anecdotal stories are difficult to deal with. We don't know what's actually happening. Turns out that what you heard is not correct. So let's get to a question of what's the possibility of bats in North America being reservoir hosts? My understanding is there aren't any in North American bats, but, they haven't been sampled all that much. We did a TWEVO a while ago with Simon Anthony, and he's done some sampling, and he said they're not in North America, but I'd like to see more sampling. Yeah, Amy asked, why have a rapid test so someone can take it before entering my house? Yeah, I think there's still a place for a rapid test and for future outbreaks as well, just to develop the technology. Yeah, and remember, death reporting can be very inaccurate depending on where it is. So you just have to be really careful. I'd like to see, the data on paper and know where it came from. The coloring books, listen to TWIV and you'll hear about them for sure. After the vaccine, do antibodies increase linearly or exponentially? Well, there's an exponential increase, and then it plateaus at about 60 days after. There's a nice preprint out of Israel showing responses to the Pfizer vaccine, and they peak in about 60 days and stay pretty high. And then they start to decline slowly. They decay over the next year or so until they're very low levels in the blood. They're still there, but they're low levels and not enough to prevent infection. You have to wait for the memory cells to kick in, and then they will make more antibody. But it happens very, very quickly. Is it possible to create an mRNA vaccine that makes antibodies to a part of the spike that won't change? Yes. People are working on that now. The head is more variable than the stem, just like for influenza virus spike. So people are working on that. I think it might work. It's going to take some time, though. It's going to take some time. Yeah, well, it's okay, Megan. Some people are interested. It's a 2-hour show. I understand if you can't listen. I have no prejudice against that. It's been fine. You've been binge watching TWIV from a few years ago. Visiting virologists. Yeah, I used to travel a lot. In 2019, I flew a ton, and the last trip was Singapore, when apparently this virus was already circulating not too far away. I used to enjoy being invited and doing TWIVs remotely in all places, Asia, Europe. Hope I can do it again. And you may ask, why do you hope you can do it again? Well, not because I think the virus is going to be a problem forever, but because I'm not sure we're going to have that kind of travel anymore for me to go somewhere when I can do it by Zoom, right? It's a little different, but it just may not happen again. We'll see. We will see. No, I don't think post-fusion contributes to blood clots. There's just no evidence for that at all. No. When the time comes, who's going to announce to us that herd immunity has been achieved? Well, it's not going to happen overnight, right? It's going to take some time to do that. So it'll happen gradually. We're going to see the numbers drop slowly, and Tony Fauci will start to talk about it probably. And then, maybe the president will announce it someday, president of different countries. Who knows? So let's see. I'm going to just, in the last few minutes here, thank some people for their super chats. Tom, thank you so much. By the way, Susan, thank you so much for your super sticker. Fernie, thank you for your super chat. This all goes to supporting Microbe TV science communication. Thank you, Wild Bunny. I'm glad that I've kept you sane. It's a pleasure to be able to do that. Thank you, Bad Cat. Could terrorists use gain of function to create new pandemics? In theory, but I don't think they could. It's really hard to do. I've been working on viruses for 40 years, and I couldn't do it, and I'm pretty damn good. But every time you mess with a virus, you mess it up. Nature's the best genetic engineer, and that's why these come from nature. Thanks, Post Polio News, for your super chat. Thank you, Wendy. Daniel Griffin and I are going to be sharing office space in Manhattan on 8th Avenue. Thank you, Susan. You're so kind. And your super chats will go towards supporting that office, and I'm going to be there almost every day trying to make science communication, podcasts and videos, so forth. Thank you again, Tom. I try to have good humor. I'm getting better. From the first days of this live stream, I was reacting poorly to everything, but I'm getting better. As someone says, you get thick skin, right? For sure. Thank you again, Susan, for your generosity. Very kind. There's a high bar in documenting repeat infections. Could this result in their being underreported? Absolutely. Totally could be underreported. But how much underreporting? Because if it were really common, we would see more evidence of it. But, time will tell. We will keep seeing what's going on. Okay, those were the super chats. So Vincent can announce when we have her. No, I have no authority. we'll talk about it on TWIV. I think we're already getting there. Here's the thing. Someone from a Pennsylvania newspaper called me today, and she said, can you help me interpret these numbers? And she gave me, where is it? Did I write it down? Yeah. Okay, so I wrote this down. So she said we have 11% of the population of Pennsylvania has had full vaccinations, 23% partially. And then she said, we have 1 million cases. And I said, well, you probably have 10 million cases. There's probably a tenfold underreporting. So you probably have 40% immunity. So you're approaching herd immunity, 50 to 70%, more like 70%. And so I said, it's good. I think it's a great sign. And I'm positive. I'm very positive. I think by the end of this year, we're going to be back to normal. Yes, I have to ask. I'm sure he will do a Q&A. Usually Daniel is pretty good about doing things that I want him. I don't overtax him, right? So I do think, and it's just a matter of whether he does it. Now 8 p.m. He usually doesn't like to do things after 5:36 or so, but we'll see what time. It may be a weird time for you folks, but I will let you all know. Of course, I'll announce it here ahead of time. You'll be the first to hear about it. And yes, I just released This Week in Neuroscience, but I wasn't on it. It's all without me because I was doing TWIV at the same time, and I couldn't do both, right? So I let them do it. And actually, they did a pretty good job, and the sound is pretty good. Is there a methodology for finding conserved epitopes? So you can do it in a number of ways. You can first look at if you have lots of spike sequences, not just SARS-CoV-2, but different coronaviruses, you can compare them and say, okay, are there epitopes that don't change as much? And you can do that with the flu HA2 spike. And then you have to make the proteins and test them in animals. Unfortunately, in the end, you always have to test the ability of the proteins to make antibodies and see if they cross-react. You have to make the spikes where the virus is carrying them and see if they cross-react. You have to do the down and dirty science. But yes, there is a way to do that. Oh, yeah. A lot of North American bats are dying off from a fungal disease. That's right. Because they hang in the caves and the fungi are in there and their immune systems are down, and so they get infected. Yeah, it's a real problem. I'm a bit lost on the rapid tests. In Europe, we can buy antigen rapid tests from the pharmacy quite cheaply. Why aren't they common? I don't know. That's a great question. I am amazed because here they say they can't even develop them. I have to look into this. Would love to get my hands on one, or I'll look into it and try and get some data. I didn't say accept. That's what she told me. And she said, what do these numbers mean if true? And what am I going to do? Tell her I don't believe them? I have no idea. But I think the 60% in Michigan is an issue if people are getting infected. Something's wrong there, or we don't know something, right? It's either in groups of people who are not infected or immunized and they're collecting together, or something else. So I don't accept it. I just told her what I thought. And I do think as a whole, this country, the numbers are going down, and it's a combination of infections, natural infections, and vaccination. That's my idea. If you have pockets of non-immunized people, you're going to have outbreaks, of course. Yes, I did a tour. Not like the Needle, though. They didn't let me inside, so it wasn't filmed. But I did a podcast. I've done a couple of podcasts at the Rocky Mountain Lab, though, for sure. Adam has been on TWIV before. We could certainly get him back. Yes, I agree. He would be an interesting guest. I thought Texas already ripped off the masks, right? Thank you, Peter, for your super chat. Very much appreciated. This is great. We have 534 people today. It's very impressive that you come here and chat with us. and amongst each other, of course. If nature is the best engineer, then why is my natural immune system not superior? Oh, well, your immune system is pretty damn good. If it weren't, you'd be dead. We'd all be dead, right? Our immune systems are amazing. The problem is viruses out-evolve us because they reproduce faster. It takes us a long time to reproduce and for selection to occur in humans, and viruses can just do it quicker. Just do it quicker. I'm not sure FDA has got issues with rapid tests. Last year I was on a call with the FDA, and they were very excited about rapid tests, and they were trying to facilitate it. So I don't know what's happening. I don't know all things, just some. Thank you, Westfield90, for your super sticker. I really appreciate it. Yes, the positive rate should be slowing down. That's what I think they're happening, but they're plateauing in some places because what happens is when people see the rates going down, they rip off their masks and they go to restaurants and they hang out. Okay? And then you're going to see a little plateau. Hopefully it goes back down again. That's very nice. The best immunity from insanity is listening to Vincent. Thank you so much. It's very kind of you, folks. Could you publish exam or quizzes? Yeah, I've been putting the quizzes up on SurveyMonkey. You can link to them in the YouTube. I'm a little behind, but I will get them all up eventually. Not many people were taking them, so I kind of fell behind the quizzes, right? You like the parasitism video? Yeah, those are very good. Between Daniel and Dixon, they're great. I will be here after normal. I'll be here talking about viruses. As long as there are a few people here, 10 people, I'll have a chat, Wednesday nights. And if there's no one here, it'd be weird to just talk on my own, right? Well, that's just a podcast. But I think people will come here and, hey, we didn't even get to talk about Ebola virus, right? Here's the thing. The Ebola outbreak in Guinea seems to be from a virus that circulated in the last outbreak. And where it went is not clear because it's only 12 bases different. And if it had circulated, it would have more changes. So it is not clear what's going on. That's very interesting. Thank you, Susan K., for your super chat. And thank you, Plinky, for your super chat. Really appreciate it. I'm glad you like these discussions. Why are health authorities not telling people to get healthy? Well, I think they do, right? But people don't listen. People want to do their own thing and chill, right? I try and stay healthy, but I don't get enough sleep. And when the pandemic is over, we should do chats in person for people who can make it. I don't know what it means in person. Isn't this in person? Oh, you mean actual? Yeah. People in New York, I'd do a live thing somewhere. It doesn't even have to be in a theater. It could be in the university. Sure. Ian Fraser. Thank you, Ian. I appreciate your generosity, and we'll put it in the studio for sure. And any of you who come to New York, you want to come to the studio, come by and we can chat for sure. Just watch videos from India and you will know why it's rising again. Zero distancing and zero masking. Really appreciate it. Thanks for this Q&A. I work on a SARS-CoV-2 test site. We're being asked to isolate and get tested if we have any vaccine side effects. I don't know what that's coming from. Are you sure? Why would they worry? Are they worried about reinfections with variants? I think that's ill-advised, frankly. I don't think that that's an issue. But could I update and upload the syllabus? So the syllabus is there with every lecture, right? Virology. Well, you can go to the YouTube and below they have links to the syllabus, right? All right. Folks, I want to thank you all for coming here. 526 of you, 240 likes. It's pretty good. We've done higher on the likes. We had 300 once. But that's fine. The liking is just so more people will listen, right? Because when they see other videos being liked, they come here. That's very nice of you. Get in touch with me. I can get you some on discount. Okay. I can get you a cheap price on sorry, I can get a cheap price on the textbook. I'm distracted by a text. All right, folks, have a good evening. Thanks for coming. I so appreciate it. I appreciate your coming. I appreciate your donations, and I appreciate your involvement. And we'll see you next week. We'll be back. Good night.